Assalamualaikum warahmatullahi wabarakatuh and a very good morning to Dr. Aida and my fellow friends. Today me and my partner from group 2 will be presenting about the company current technology and the improvement that Lablink can make to advance further in the industry. Our main focus is PCR polymerase chain reaction because Lablink has been using PCR to run many kind of tests. So what is PCR? PCR is a method used to make millions to billions of copies of a specific DNA sample, which allows scientists to take a very small sample of DNA and amplify it to a large enough amount to study in detail. In this presentation, we will be focusing more on real-time PCR. Real-time PCR is a molecular biology technique used to monitor the amplification of the target DNA or RNA sequence during the PCR in real time. So what is the difference between PCR and real-time PCR? Conventional PCR can only be visualized by running amplified DNA samples through electrophoresis to detect the presence or absence of DNA amplification. Meanwhile, in RT-PCR, the amplified DNA is labeled usually with fluorescent dyes, enable the amplification of DNA visually by reading the fluorescence of the amplified DNA in a real-time manner. In Lablink, RT-PCR has been used for COVID-19 detection, Zika virus detection, meningitis viral pathogen detection, and many more. Before I go further, I will explain a little bit about the principle of real-time PCR. Real-time PCR is a process that is conducted in a real time. The amplification of DNA is linked to the generation of fluorescence, which can simply be detected with a camera during each PCR cycle. The principle of real-time PCR relies fully on the use of fluorescent dye. Hence, as the number of gene copies increases during the reaction, so does the fluorescence, indicating the progress of the reaction. The next slide is how does RT-PCR work with COVID-19 virus? A sample is collected from the parts of the body where the COVID-19 virus gathers, such as from the nose. The sample will then be treated with chemical solution to remove all the unwanted substances so that only the RNA is present in the sample. The RNA is then being reverse transcribed to DNA using reverse transcriptase. Scientists will then add additional short fragments of DNA that are complementary to specific parts of the transcribed viral DNA. When a virus is present in the sample, this fragment will attach themselves to the target section of viral DNA. The third step is the mixture is then placed in an RT-PCR machine. The mixture will go through cycles in different temperatures to create new identical copies of target section of viral DNA. The next step is as new copies of the viral DNA sections are built, the marker labels attached to the DNA strands and then release a fluorescence dye, which can be measured by the computer machine and will be presented on the screen in a real time. The last step is the computer will track the amount of fluorescence in the sample after each cycle. When a certain level of fluorescence is surpassed, this confirms that the virus is present. There are many advantages and disadvantages of real-time PCR. The advantages are they are cost-effective, time-efficient, more sensitivity and specificity, increased dynamic range of detection, and no post-PCR processing. Meanwhile, the disadvantages of real-time PCR, the kits are not available for all kinds of genes and disorders, and cross-contamination between nucleic acid can occur.
So I will continue with the improvement that can be made by Lablink, which is next generation sequencing. NGS refers to technique in which millions of nucleotide sequences are distributed simultaneously and also known as a high throughput or massively parallel sequencing for DNA, RNA, and methylation sequencing. The principle behind NGS is similar to Sanger sequencing, which relies on capillary electrophoresis, but it evolved from traditional fragment-based Sanger DNA to array-based sequencing. NGS allows to rapidly sequence whole genome, deeply sequence target region, utilize RNA sequencing to discover novel RNA variants and splice site, or quantify mRNA for gene expression analysis, analyze epigenetic factors such as genome-wide DNA methylation and DNA protein interaction, sequence cancer sample to study rare somatic variants and tumor subclones, study human microbiome, and identify novel pathogen. NGS is divided into two generations, which are second generation and third generation. Second generation is a short read sequencing method, which require less than 2 to 400 base pair. It is cheaper to produce, which use less reagent and less cycle, and also completed when there is a reference genome. While the third generation is a long-read sequencing method, which require more than 400 base pair. It's provide more sequence that can be used to assemble genome or transcriptome. This slide shows the NGS workflow in clinical pathology. This shows the standard workflow for NGS assay. There are numerous steps involved which require technical precision. The steps may vary depending on type of assay and the information required. After the data analysis, there are validation reaction and clinical decision step. This is to confirm which treatment that can be used to treat patient. There is plethora application of NGS in clinical, which are mutation detection in Mendelian disease, recognizing disease predisposing and protective factor, improve diagnosis and treatment of cancer, pharmacogenomics, identification of structural variants, epigenetic and sequencing cell-free DNA fragment. Next is the advantages and disadvantages of NGS. For the advantages, it's provide higher sensitivity to detect low frequency variants, faster turnaround time for high sample volumes, comprehensive genomic coverage, lower limit detection, high throughput with sample multiplexings, and ability to sequence hundreds to thousands genes or genes regions simultaneously. While for the disadvantages, it is a short reading sequence, it challenges in assembly of the reads, complicated data analysis, and large data storage required. So I will continue with the comparison between RT-PCR and NGS. For the pros of RT-PCR, it has a family workflow, capital equipment that already placed in a most lab, and good analytic sensitivity. While for the NGS, it has higher discovery power, higher sample throughput, and good analytic sensitivity. While for the cons of RT-PCR, it can only interrogate limited set of variants, virtually no discovery power, and less scalability. While for the NGS, it has less cost-effective and time-consuming for sequencing, low numbers of targets, which from 1 to 20 targets, and also it has complex bioinformatics. So this is the reference that we use to refer. So I think that's it from us. Thank you. Is there any questions?